So next, it's my pleasure to introduce Ben Brown. He is from the Lawrence Berkeley National Lab in California and the University of Birmingham in the UK. He's going to share with us a multi-omics strategy for fly models of human diseases in exposure biology.
Thanks very much. Thanks to the moderators. So I'm going to talk about trying to develop some strategies for building new sorts of models in exposure biology. So what is exposure biology and what's the deal? There's a long history of Drosophila as a model in exposure biology, and what we're trying to do is maybe trying to expand that a tiny bit. So in toxicology and exposure biology, generally concerned with environmental chemicals, and there's a tremendous amount of chemicals in our environment. There's over 100,000 chemicals used by industry, and about 93% of them have absolutely no toxicological data, so we don't know what they do to an organism. Some of them have very pervasive environmental distributions, like the pesticide, the herbicide atrazine. 33 million Americans drink atrazine-contaminated water. In the Midwest, particularly well water, can get some pretty impressive levels. And so why is there this dearth of toxicological data for chemicals? It's because traditional testing is very slow. It takes 5 years and costs about $1.5 million a pop, and generally uses 1 or 2 rodent models. So we know at the end whether or not it's lethal to a rodent, whether or not it causes cancer. This has created about a 30-year lag in regulation of chemicals in the environment. Between when the industry innovates and government finally regulates, usually through the collection of epidemiological data. Asbestos, for instance, was not just in insulation, it was in cigarettes. You just inhale it directly into your lungs and skip the middleman. And that went on for a very long time. There's lots of other examples, and we're still doing it. Nothing's changed since the 30s, 40s, 50s, 60s, 70s, or not much. The carbon nanotubes are now a widespread environmental contaminant. They cause necrotic lesions in the lungs. Probably not cancer like asbestos, because they're actually necrotic lesions, not pro-inflammatory. But they're $250 a kilo online. If you've got a Visa card, we can go online right now and buy a whole bunch of them. Just throw it up in the air. It's fine. There's no law. So we need more cost-effective models to try and change this. We need to be able to assess compounds faster on timescales relevant to industry, relevant to innovation. So it really is a grand challenge before us to try and replace this paradigm of retrospective regulation with prospective bioassessment. And that's going to require a much better molecular understanding of what environmental chemicals do to biological systems. And that's not an easy thing. The types and numbers of genes that respond to and metabolize environmental chemicals differ radically between species, certainly, but even between individuals within a species. In this room, we likely have threefold copy number variation at a variety of cytochrome P450s that render some of us very susceptible to hepatotoxicity from acetaminophen and some of us extremely resistant. So that's a pretty fundamentally tough problem. And it's actually worse than that. It's not just the genotype of the individual. Every environmental exposure, whether it's a collateral food exposure to pesticide or something you're breathing in, is mediated by the microbes that inhabit your body. Each of us carries between 1 and 6 pounds of microbes in our guts. We have about 20,000 genes in our nuclear genome. Our microbes contain about 1 million. And while those are organized into orthologous groups, they still pack a whole lot of enzymatic potential. So what we're trying to do is understand how to complete this picture, how to understand the impacts of genetics and microbial content in the processing of environmental compounds. The model system that we're working on is pesticide exposure. Pesticide exposure is a lovely model, extremely prevalent in the environment. I already gave the example of atrazine. They constitute endocrine disruptors. Many of them generate neurological phenotypes. Paraquat, for instance, is a Parkin mimic. In fruit fly, also causes Parkinson's-like symptoms in humans. Atrazine causes liver disease. And current safe doses for these compounds are more or less guesstimated from really high-dose studies, mostly in rodents. And low-dose daily exposure is extremely common. Worldwide, there are over 25 million hospitalizations for pesticide exposure every year. In the U.S., it's cost more than $1 billion a year, estimated, most of that due to unanticipated cancer. And furthermore, interactions between pesticide and the microbiome are very poorly understood. When you are exposed to a pesticide, that thing is very often rapidly metabolized, and your microbiome can metabolize it into stuff that's much less toxic or much more toxic. And of course, that depends on the details of the compound and your microbiome. So here's the initial study design I'm going to talk to you about today. It's actually two species. It's fruit fly and mouse. And I'm going to focus almost entirely on the fruit fly study and just to tell you a little bit about the mouse at the end. It's a chronic exposure scenario, a couple of days in fruit flies and a couple of weeks in mice. And in both cases, the doses are pretty low. The fruit fly is consistent with agricultural exposure, so think what a honeybee might experience, while the mouse is consistent with Midwestern well water. So, as I said, paraquat induces a nice locomotor phenotype. I'm going to show you a video of fruit flies being exposed to, or who have just been exposed for a couple days to atrazine, and they also have a locomotor phenotype we were delighted to see. So each of these flies is a different color because they're being automatically tracked by an algorithm. And you'll see this green guy right here do a nice little spin, a nice little twirl. It's a locomotor phenotype we don't see in the wild type version of these flies, but it's very acutely present in the atrazine-treated flies. Here is the linearized genome of the fruit fly responding to this low-dose atrazine exposure. So each of the chromosomes is lined up end to end here along the x-axis. The y-axis is fold change. Every gene is represented on this plot. If a gene spikes up, it's induced. If it spikes down, it's repressed. And you see a lot of stuff that we usually expect to see with toxic exposures. We see the suppression of genes involved in germ cell proliferation. We see the suppression of a lot of digestive enzymes. We see some interesting effects for genes involved in lipid synthesis, lipid localization, lipid processing. Some of those are listed down there on the bottom left. But interestingly and notably, the fruit fly does not have any genes in its genome that can process atrazine. And so actually for the duration of the exposure, we would have expected to see more acute toxicity. This does not look, if you study, cadmium or arsenic or high-dose toxicants, this does not look like a really acute toxic exposure. You don't see a huge heat shock response, et cetera. So why is that, since the fruit fly can't process atrazine? Well, we look to the fruit fly's microbiome. So Sue Selnicker's group has been working for a while, one of several groups working to map the fruit fly gut microbiome. And how do I go back? And this shows the fruit blast. Sorry. This shows the fruit fly gut microbiome evolving over 72 hours in the adult fly. And as you see, the microbial content changes and evolves and diversifies a little bit. Sue's managed to capture and culture at least one representative from each major clade in the fruit fly gut microbiome for the flies in her lab. And resequence those using PacBio and Oxford Nanopore technology into single contig bacterial chromosomes. And when you do this PacBio, this is a Acetobacter tropicalis. It's used widely in industry. It's been sequenced to Illumina to many thousands of X-fold. When you do the PacBio, she got 1,000 new genes doing PacBio as opposed to Illumina. So it really does benefit to use those third-generation sequencing technologies. When we look at how the gut microbiome changes over time in control versus pesticide exposure, here showing paraquat, we see that there are substantial effects. We see some microbiome remodeling. In particular, paraquat, by 48 hours, has really started to eliminate the Acetobacter from the fly gut. And by 72 hours, it's pretty much completely gone. Lactobacillus and a few others have taken over the metabolic niche that it previously occupied. And the same is true for atrazine. On this plot down here, we're plotting paraquat versus atrazine, the fold change. And Acetobacter is depressed to about 25% of its wild type level by 72 hours in atrazine, whereas it's completely annihilated in paraquat. Interestingly, in vitro in culture experiments show that paraquat is toxic to Acetobacter, whereas atrazine is not. In culture, atrazine has absolutely no effect at any soluble dose on Acetobacter, but in the gut, it changes the competition landscape or does something, or some secondary metabolite that makes it much less competitive. Here's a summary of the fecal metabolome. This is just a high-level multivariate study because monovariate statistics on the fly fecal metabolome are pretty tricky. Fly poos are about a nanoliter, and that is not an easy metabolomics problem to solve. Mark Viant, University of Birmingham, got this together, and it's still a work in progress. But so far, it looks like we've seen some interesting changes driving this in lipid metabolism, and I'll be able to show you that more clearly in mouse in a moment. So now back to the hunt for genes involved in atrazine metabolism in the fly gut. So we looked at the genic content of all of these clades sequenced, and their pathway by which atrazine is metabolized by through an aerobic pathway is very well known, and we look for those genes, we see them immediately, shown down here. Notably, the entire pathway is not present in any one microbe, but it is present in a consortium of microbes in the fly gut, and that explains why, going back to this metabolomics, one thing we did check very, very thoroughly in this work was that there is absolutely no atrazine nor any of its secondary metabolites in the fly feces. And that's presumably because this microbial consortia is actively metabolizing all the pesticide. We also find all of these other genes all of these other enzymes that are biochemically related to these atrazine metabolizers, but these are more or less new to science, right? I mean, they're related to enzymes that we know about, but who knows what the various substitutions have done to their preference or their activity. Now, for a little bit about mice, to try and link this a bit closer to human disease. So mice also undergo microbiome remodeling in exposure. So shown here are summarizing atrazine and paraquat exposures in two different strains of mice, AJ and C57. For every pesticide to which we've exposed mice so far, atrazine, glyphosate, paraquat, we see a drop in rotarod performance, a slight drop in coordination, reflective or reminiscent of what we see in fruit flies. When we look at the content of the microbiome, between comparing sham to exposure situations, we see a nice gene-environment interaction. The collection of microbes that are depleted or enriched after pesticide exposure is dependent upon the genotype of the mouse. So it's different in C57 than it is in AJ. In fact, it's very different. So the effects of the pesticide on the content of the gut microbiome have a genic component. When we look at the mouse cecal metabolome, we've looked at the mouse cecal metabolome for all these. Mice are very nice in the sense that they have an enormous amount of material. Their poos are huge, and they have a nice ample cecum. So the metabolomics is much higher resolution. Here's a summary just showing that the male and female atrazine separates out nicely from the controls that are next to it, and then from the paraquat that's on top there. And when we do a dimension reduction analysis to try and identify what drives this, we find a nice cohort of glycerophospholipids, which hopefully is what we'll ultimately end up finding something pretty similar to this in the fly too. But here's the healthy control mice right here. We see some male versus female difference. This is male is female. Difference in the natural content of glycerophospholipids in the cecum. And then we see a shift, a depletion of lots of stuff that's usually there, and an enrichment for lots of stuff that's usually not. And the response to paraquat is overlapping, but also quite distinct and also sex-dependent. So glycerophospholipids are biomarkers for a number of human diseases, including liver disease and arterial sclerosis. So the take-home here is that by looking carefully at the molecular response to exposures, we might be able to start to map responses in a fly to human disease-relevant modes of action. So with the team, thanks so much. Susan Selnecker leads this project, and then we've got a big crowd at LBNL. And University of Birmingham, Mark Vine did the metabolomics in fly, and then University of Oklahoma, Vincent Bonifay did the metabolomics in mouse. Thanks so much.
We're going to have one question up here.
So that was very interesting. So tell me more about, like, did you feed flies antibiotics to show that the microbiome is really causing resistance to the chemical? Or?
So the nodobiotic experiments, right, to show that the microbiome is what's mediating the lack of toxicity. Right. That's in progress right now, so we don't know yet.
So should I keep drinking Midwest well water or not?
I would not drink Midwestern well water if I were you. This is set up, a really natural epidemiological experiment now, right? We'd like to see if Midwestern residents near agricultural lands have reduced neuromuscular coordination. Because the mouse dose is very, very low, and those guys are falling off the rotarod noticeably slower than, at noticeably slower speeds than other mice.
Actually, along those same lines, the fly microbiome that can break down atrazine, have you tried transplanting or introducing that into your mice and then showing that it's not absorbing the atrazine anymore?
The mice have microbes that are compatible with that pathway, absolutely. The problem is it is an aerobic pathway. Anaerobic metabolism of atrazine has been demonstrated, but no one has mapped the pathway.
Thank you so much. Okay.